you here know me already. I'm a research fellow working with the Berrimah Vet Lab. I came here to Darwin in June last year and have been working on this. I'm very happy to share with you this morning some of the data we have so far collected on this project. Perhaps to say a few words about the title of this presentation. You can see from what was sent around, I've modified it and added tissue tropism. Tissue tropism, we are looking at the different tissues where the virus might be found, because previously this virus has been known only to appear in blood, and now we are trying to make efforts to show that it can also be found in other tissues. And the immunohistochemical part, this was a technique we've standardized and validated for demonstration of viral proteins in tissues of infected animals. So I'll probably go ahead with the introductory slide. Bovine ephemeral fever is a viral disease caused by an RNA virus. This virus belongs to the group called rhabdoviruses. They are transmitted, in this case, by mosquitoes. There are suspicions also that other biting flies like cricoides can transmit this virus. In the family of these viruses, we have the rabies virus and the Barigye virus here in Australia, which are not known to be transmitted by arthropod vectors. But we have related virus to the bovine ephemeral fever virus, particularly here in the NT region, that are transmitted by mosquitoes. And the disease it causes is typically characterized by a fever, very short-lived fever, and a short-lived viraemia. Viraemia means the presence of the virus in the blood. The longest we have been to detect the virus in the blood is about 5 days, but most of the cases it will just appear for a day or so and then disappear. The other symptoms would include loss of appetite. These animals will be seen not to be feeding. And the other constant feature of the disease is lameness. You find these animals limping around. And paresis. In a few of the animals, about 1%, they kind of get paralyzed. And there is a basis for this paresis. What is known during the acute phase of the disease, there is a sudden drop in blood calcium that kind of paralyzes the muscles. I have a video that doesn't seem to work on this system that I thought would share and show the kind of clinical symptom I'm talking about here. And because this disease is short-lived, the presence of the virus is short-lived, the diagnosis sometimes is problematic, because if you don't get there in time when the virus is present in blood, then you are going to have to rely on taking serum samples and demonstrating the antibody against the virus. Usually this one takes much longer, and we needed to do something about that to get, probably another method of demonstrating the presence of the virus to be able to say that this animal has been infected. So the objective of this project, number one, we needed to develop an immunoperoxidase staining method. This is a unique and kind of elaborate staining method that can be used to stain for viral proteins in tissues. The advantage with this technique is that you can show specific cell types and specific tissues where the virus is present. And we're so fortunate that there is a group in Queensland that developed a monoclonal antibody. Monoclonal antibody can be used as a probe to specifically pinpoint where the viral proteins are. So that was our objective number one. And this technique has potential for application in research into this disease. And in the future, of course, it can be developed and further enhanced for diagnostics of bovine ephemeral fever. Objective number two was having now standardized this technique, use it alongside polymerase chain reaction, which the Berrimah Vet Lab is already using so efficiently, and study the viral tropism. So we've used these two techniques to study the distribution of the virus in different places. And the third objective, which we are just about to embark on, is to demonstrate what is behind the inflammation that causes the limping that I was talking about, that causes the sudden increase in neutrophils in the blood, neutrophilia, and also study a little bit about the immune response. We know that these animals, after infection, after they get solid immunity, they are protected. But the basis of that immunity is not known. And I think to be able to design better vaccines, we need to know a little more about what constitutes the protective immune response. Specifically speaking, we shall be looking at the pro-inflammatory cytokines that the drivers of all these symptoms and the pathogenesis of disease during the acute phase of the disease. And as you can see, we shall be testing for 7 cytokines. We have interleukin-1 beta and tumor necrosis factor, interleukin-6. These are the most crucial in inducing fever and inducing inflammation because, like, as I said, this disease is typically an inflammatory disease. Then we have another set of cytokines or growth factors, granulocyte colony stimulating factor and granulocyte monocyte colony stimulating factor that are entirely responsible for stimulating the bone marrow to start production of neutrophils. Because when there is an inflammatory crisis, the neutrophils circulating in the blood are not enough, so you need to manufacture more and push them out into the circulation so you can be able to control the infection. So I included these 2 down here. Interleukin-10 is kind of antagonistic to the inflammatory response. So if we can look at interleukin-10 alongside these other ones, we shall be able to see the kinetics of what is going on in the system. And then interferon gamma is very crucial in initiating not only the inflammatory response, but also the immune response. So towards the end of the acute disease, I hope that we shall see an increase in interferon gamma. Then as far as the immunological response is concerned, my view is that we should look at lymphocyte responses, specifically CD4 cells that actually are responsible for mounting, for initiating the immune response. And the CD8s are always there in viral infections, excuse me. So we need to look at the responses of those cells and kind of have an idea what is going on at the beginning of the immune response. And the last objective is to compare the sensitivity of PCR and the virus isolation in the diagnosis of the disease. We're already collecting data on this to be able to compare those 2 techniques as diagnostic methods for BEF. So I'll start with the strategy that we had to kind of address some of these objectives. And the first thing we had to do with objective 1 to develop the immunoperoxidase staining method was to kind of find a positive control for our assays. And because no one had any tissue known to be positive for the virus, we had kind of to improvise. Steve helped us, grow these cells, infect them in the lab, and then we could use them. Here is a schematic that explains it all. You grow the virus in these cells. They are hamster-killing cells. Around day 3, you harvest these cells before the virus starts, splitting these cells and rupturing them. You harvest the cells, spin them down, and have a sediment, a small sediment of cells. You use a bit of that to test with PCR and make sure that the virus is there. And then the remainder, you actually embed it in paraffin. And now you have you improvise kind of a tissue that is positive with the virus. Then you can use it to look for the virus in bovine tissues. So here's the results. This is a very interesting slide. I didn't show it before, but this is the very first time we tried this assay. And here what we have are those cells infected with the virus that have been probed with this monoclonal antibody DB5 that is highly specific for this virus. In fact, highly specific for a specific viral protein. Even at this dilution of 1 in 200, the first time I saw this, I just jumped. What you see here, these are just cells, but you can see kind of granular appearance. Compared to these are the same cells probed with just mouse serum that doesn't have the specific antibody against the viral protein. And what we can see is just antigen, viral protein reacting this way. This was beautiful. Now, after getting this result, you need to kind of get a working dilution for your antibody because you don't want the assay I mean the result look like this, then you are going to have what they call noise, background noise. So you want it at a good dilution. So you have to do a series of dilutions for your antibody and get working dilution. Now, this is the slide of non-infected cells. There was a stark difference. If you look at the non-infected cells, you don't see that granular appearance around the cells. And of course, these were probed with the monoclonal antibody, and these were also probed with the mouse serum. So, as we walked through the dilutions, this was the final result that we got. Here you have virus-infected cells probed with this monoclonal antibody at a dilution of 1 in 1,000. And what you see here are multiple cells that have this kind of brown to reddish coloration within the cytoplasm. That is nothing but viral protein, because given the high specificity of this monoclonal antibody, what you are looking at there is just cells loaded with virus. And on this side, the same cells infected, but probed with negative mouse serum, and you don't see the same response. So to make sure that what we are seeing here is a BF virus, we also had cells infected with non-related viruses, including blue tongue, acaban, and so forth, and we also tested them, and indeed there was no positive reaction. So then once we had this block of virus-infected cells, now we could go ahead and test bovine tissues. So here is a schematic of what we've been doing. We have relied on naturally infected animals that we get in the field. Here within the NT, we collect all sorts of samples to do all sorts of studies that we are doing, but specifically how it relates to the immunohistochemistry. Those animals that die or that are euthanized, we harvest tissues and body fluids, and those tissues, the fresh ones, are tested with PCR to see which tissues have the virus. And as it were, some of the tissues, we fix them in 10% formalin, and then go ahead and do immunohistochemistry using our new method, and also do routine histopathology just to see and characterize any pathology that is there. We also collect blood and serum and do all sorts of other studies that I'll be talking about. So some results here. When I arrived here in June last year, there had been an animal that had been diagnosed in Alice Springs with bovine ephemeral fever, and these are some pictures of that animal. This was in April before actually I arrived here. And this animal had acute disease. It was confirmed to have had BEF based on serology. But this animal never really recovered. It recovered from the acute disease, but still had residual neurological symptoms. If you saw this animal, even when I went there in July, it still, when it walked, you could see there was something wrong with its nervous system. But anyway, to emphasize the clinical symptoms you can see this animal looking very dejected, was not getting up. I think someone is trying to give it some water to drink so it can't die from dehydration. And this is a video that I didn't succeed, the system can't show it, but this animal was, it literally could not get up. And eventually it did get up, but you could see it staggering. And when we did the histopathology, some of the tissues that we harvested in July showed some of these lesions. These lesions had been reported as far back as 1970. This is skeletal muscle from the thigh, and you could see these lesions. These are areas of inflammation where the muscle fibers have been fragmented, and you have macrophages, a specific type of white blood cell coming in to remove the dead tissue. And then you have similar foci in the heart. I mean, if you have a heart with areas like this, then anytime you could die from heart attack. So again, you have fragmentation of heart muscle with the macrophages coming in to mop up dead cells. Not very far from this area, there was a small blood vessel that showed inflammation around it. And again, I include this picture because a lot has been talked about inflammation around blood vessels in bovine ephemeral fever. So when we did immunohistochemistry with our new method, this is a picture of the spleen. This is what we saw. Okay, do you mind dimming the lights? Yeah, this was very exciting. All over the spleen, but this shows just on the surface, there were these cells that were picking the new stain. And there were two populations of cells. This particular cell you see here, showed with a short arrowhead, and then these spindloid-shaped cells around blood vessels. Cells that look like this are called parasites. They're usually found around blood vessels, and they invest the blood vessel, and evolution has provided that as a kind of protection. If things spill over from the blood vessels, they're usually trapped and invested beneath those cells. But these cells, when we looked much closer, they looked like macrophages, a specific type of white blood cell that has very important roles in fighting infection, but also has some roles in mounting an immune response. On this side, you have the negative control probed with mouse serum. Here is, of course, our monoclonal antibody as the probe. Now, something so remarkable about this animal is that this was 93 days from the time acute BEF was diagnosed. I thought that was remarkable because the only time that we knew the virus could be detected in an infected animal was that window during the acute phase of the disease for at most 5 days during the acute disease. So here we hadn't tested this spleen by PCR to confirm for real if the virus was there. And as I will be showing, of course, eventually we're able to use PCR to show that the virus was there, or at least a fragment of the virus in some form was there and was detectable by PCR, which is a highly sensitive technique. This is another area of the spleen towards the center. All you are seeing, these blue cells are lymphocytes, and lymphocytes are so crucial in mounting immune responses. This is where you have the CD4 cells, the CD8 cells, and so forth. And there are these small blood vessels, and just around them you could see these cells, which morphologically are consistent with macrophages, kind of containing viral protein. And we know that if you look at the spleen, the function of the spleen, it's so crucial in mounting immune responses. And then here you have macrophages, which are known to process viral protein and present it to lymphocytes, thereby beginning the immune response. And when we looked at the lung of this animal, same animal, we saw these cells just on the surface. This is called the pleura, which is just a thin lining on the surface of the lungs. And you had these cells which kind of have appendages, and they are morphologically they are consistent with dendritic cells, again, which are highly specialized cells in processing antigen and presenting it to lymphocytes as the initial step in mounting an immune response. Of course, when you got into the interior of the lung, you saw all these cells everywhere, and particularly around the blood vessels. On the other hand, on the negative control, you don't see anything. Now, I thought what was remarkable with this finding again is Dr. Young in Queensland in the 70s and early 80s perhaps had reported using immunofluorescent antibody tests that he had located viral protein in these cells, which are called mesothelial cells. They just line the surface of these organs, both in the liver, lung, and other organs. And here what we were able to show using this test is that actually you have cells just beneath. And now we might wonder why these cells are there. We know that in the BF there is outpouring of fluids into the body cavities, and those fluids we have already demonstrated that actually they contain a virus during the acute phase of the disease. So these cells, we know they use their tentacles to reach out into the body fluids, and antigen that has absorbed on the surface of these organs, sample those viral proteins, take them inside, and they will migrate to the spleen, which is like the powerhouse of mounting immune responses, so to say. So I thought this was a remarkable finding. And what was also interesting is, never before reported, was when I looked at the brains of these animals, there was inflammation around small blood vessels. And this tends to happen in one specific area. I've seen it in the cerebrum, but most of the time it's in the brainstem. What you are seeing here is a small blood vessel with inflammation, different types of inflammatory cells. And you have these few cells which look like macrophages containing a viral protein. And if you really look hard enough from a pathological point of view, there is already tissue destruction here. And this is an animal that had neurological symptoms. I was just looking at the slides from one of the animals we necropsy this week, and I just found the same lesion. So for sure, this is, I think, a consistent finding, never before reported. And here I've included actually a negative control. This was an animal that died from a bacterial disease, totally unrelated to BEF. And we got these tissues from Victoria because we are concerned that because there are so many rhabdoviruses that might be related to BEF, we might not get a good negative control bovine tissue. And here, I mean, you can see areas of inflammation and you don't see any positive staining whatsoever in those areas. And of course, here we are using the same probe, this monoclonal antibody. Now again, the brains of these animals, this was quite remarkable. I just need to see more cases like this, and it will be a very significant finding. You can see this is a blood vessel again in the brainstem, a specific area of the brain, and there is this kind of staining around the blood vessel. When I met St. George, who is one of the guys that have really studied this disease, I met him last year in Gold Coast, and this is what he was telling me about. I mean, Young, who studied BEF in the high 80s, had reported this using specific stains. He showed that in BEF-affected animals, there is actually outpouring of fluids, and his suspect was that viral proteins that have complexed with antibodies actually accumulate in these areas. And here we can see these parasites kind of still investing this viral protein and kind of limiting it in one area. But sooner than later, I believe what happens is that this protein attracts inflammatory cells, and once you have inflammatory cells in the area, they start releasing enzymes, they start releasing other toxic substances that contribute to the damage of the tissue. What we have here is called a blood-brain barrier, again, an evolutionary structure that kind of restricts the damage in the brain around the blood vessels. If things can spill over from the blood vessel, let them be trapped here. Of course, if this system is overwhelmed, there is going to be a rupture of the barrier, and then there is going to be spillage of these substances into the brain. So, yeah, we were still now looking at these sections stained with the antibody, and we needed something more to corroborate what we was seeing. So all these tissues now, the fresh tissues I talked about that we collected, we went back and tested them by PCR. During the acute phase of the disease, when you test these tissues and body fluids, almost everything tests positive. As a way of explaining what's going on here, anything that's above this line, horizontal line, you consider it positive. So all these are different tissues and fluids, body fluids, pericardial fluids from around the heart, fluids from the joints, and so forth. They contain the virus. With only one exception, we were very surprised to find that the lymph node didn't test positive. We're still thinking about what's going on here. Definitely we are collecting more data to see if it is consistent that the lymph node is not positive. I don't know how it would not be positive, but yeah, we are just waiting to see. Now, another animal that died 23 days after the acute disease was diagnosed, we found that the spleen, humanode, and skeletal muscle were positive by PCR. Again, this is very interesting because the virus had never been detected in the animal beyond at least 5 days. And when we take it to 93 days in that animal in Alice Springs, the spleen, which is positive for this viral protein, is also positive for viral RNA, which I think seals this case. Definitely the virus is there in whatever form or shape, and it is detectable by those two methods. So here is a summary of the animals we have so far looked at. We've got two additional animals that are not included here, and this week we are going to get more results. But this is what I was explaining in all those other slides. So we have animal 1, 2, 3, 4, and these are the days when they were necropsed after the acute disease was diagnosed. And here is what you see. Of course, at day 1, you had everything testing positive, including blood, except for the lymph node. day 23, you had the blood testing negative, but the spleen and hemorrhoid testing positive, meaning the virus has disappeared from blood, but is still somewhere else, in this case, spleen and hemorrhoid. And day 93, you have the blood negative, of course, but spleen positive, and the rest of the samples negative. And this animal, we didn't have fresh tissues. The reason being, this again is an animal that was done, I think, in 2010 when I had not arrived. It had been diagnosed with BF. What I did was to go back to the tissue blocks and do the immunohistochemistry, demonstrate viral protein. But since we didn't have fresh tissue, we couldn't do the PCR. So I'm going to kind of summarize what we are planning to do as far as the other objectives are concerned. With objective 3, looking at specific inflammatory responses associated with this disease, we intend to test for these cytokines. And as I talk, we have everything laid out, and I think this week I'm going to start testing for some of these cytokines. We collected plasma samples from different groups of animals in the most recent BF epidemic here on the farm, and we are going to test those by antigen capture ELISA, and we are going to demonstrate the levels or amounts of these different cytokines that are present in those plasma samples. And as far as the immunological response, this will be the last objective to be tested. We shall collect lymphocytes from peripheral blood from 2 different groups, one with prior infection with BF and another one that has never been infected, and then we can compare those two, grow these lymphocytes in the presence of viral protein. I've already contacted Dr. Peter Walker. I think he did a lot of work in purifying viral protein. He's going to provide us that. That's good news. And once we stimulate those lymphocytes in vitro, then we can look at how they behave, and we can look at the percentages of populations of cells that are responding to the viral protein. And then we can also go back to the culture supernatants and test for the cytokines that they are producing. Now, to be sure that this strategy will work, what we did with one of our animals here on the farm that had prior exposure to the virus, we needed to kind of stimulate it and make sure that once we put in a virus, it can remember it immunologically and mount the response. So here is an animal exposed back last year, naturally infected around April of last year. And these animals typically, when they naturally infected, they mount an antibody response, and this antibody response reaches a peak and then it comes down, but it never goes down. It kind of stabilizes at a certain level that is protective. Now, around, was it May, I think, we stimulated this animal by inoculating it with culture-grown virus that Steve grows in the lab. And here is what happened. As soon as we put in the virus, we had this sudden increase in the antibody response. And our strategy is to kind of do the immunological assays around that time where the antibody response is rising, so we can see which cells are responsible for mounting that response. And Pleura included this picture of the spleen to show that I strongly believe that the spleen has a role in what is going on here, because we know now that there are cells there that show viral protein, and they are not there just for randomly, but they are interacting with other cells and constituting or mounting an immune response as a result. So objective 4, we are already collecting that data. We just had a recent outbreak, and each of those samples that we collected, the blood samples, definitely they have been tested by PCR. Steve is doing virus isolation, and then we also do PCR on the cell cultures, whether the virus has been isolated or not, to see and compare those different systems, which one is more sensitive for diagnosing the virus. So to summarize our preliminary findings, so far we have had 3 animals test PCR positive on the spleen on day 1, 23, and 93, like I was showing in my results. We have also had 2 animals test positive by PCR on the hemorrhoid. The hemorrhoid is a small structure which histologically looks like the spleen. So I think there is some consistency here that if the spleen can be positive, the hemorrhoid is likely to be positive. So And the cells that are positive for the viral protein look like macrophages, dendritic cells, which is another family of white blood cells, highly specialized for presenting pathogen or viral protein to lymphocytes. And then the other cells, which look like pericytes that I showed that are around the blood vessels. Now, the other interesting finding is that we saw viral protein around blood vessels. And some of that, of course, was within those macrophages, but the other was outside macrophages. And that is significant, as I'm going to be discussing. Because this is preliminary data, I'll put it as conclusions and conjectures. We shall be able to conclude for sure once we collect all the data that we are working on. Going back to our spleen and hemonode, at this point, at least we can confidently say that the virus persists beyond the acute phase of the disease in the spleen and hemonode. There is no doubt about it based on the assays that have been done and demonstrated both viral RNA and the viral protein. It does stay longer in the spleen and other tissues. And I believe that the presence of those cells that we saw in the spleen might contribute to the persistence of this antibody response I talked about, where in natural infections you have a rise in antibody titers. They come down about reach a plateau and stay there for over a year at least from the data that I've looked at. There are many other ways this could happen, but I think it makes a lot of sense that these cells which contain viral protein are continuing to interact with the lymphocytes, and of course eventually going to have activation of the B cells and then produce plasma cells that produce the antibodies. And if this method can be worked on and improved, I strongly believe it could be an alternative or an adjunct tool for diagnosing the disease, particularly in those cases where you have not been lucky to be there in a time and collect blood, perhaps to collect the serum samples. If you have a vet working in the field and all he can get you are tissues and you suspect that this might be the disease, you can go back and use this method to demonstrate presence of the virus or the virus having been the cause of death. And probably before I leave that, I didn't include a slide. That slide we saw of cell culture grown cells with viral protein, I think it's also important to remember that people who are involved in producing tissue culture vaccine, they can use that method for qualitative analysis of their vaccines. If the vaccine is not working, they can go back to that batch of cells, stain them and see if the viral protein is still there and intact. Now, I cite Young because he did a lot of work and came up with a hypothesis. He did some good experiments and reached a hypothesis in which he said that the presence of viral protein around blood vessels actually sparks off an inflammatory response that is going to damage not only the blood vessels but also the tissues around them. And seeing this in our slides and seeing another stage of the lesion kind of corroborates what he hypothesized. This is the last slide. Definitely, as you can see, this work, it's been teamwork. There have been many people that have been involved with the project listed here, Neville, Steve, and all the others that have played a role in collecting all this data. They continue to participate in the project, and we are very proud as a team to have been able to do this. And my acknowledgements to Dr. Lona Merville, who in the first place thought about this project, that has provided me an opportunity to do this work, and the funders of the project, the Meat Livestock Australia, who have provided the resources. I think the reason that BEF hadn't been really started to this depth is because probably the government wasn't allocating enough resources to study the disease. And I'm very proud that we've been able to do that with the funding from these funders. Thank you very much, and if you have any questions,